My name is Austin Graves, and I'm a postdoc in Rick Hugoner's lab in the Johns Hopkins Department of Neuroscience, along with my collaborators in biomedical engineering in the Kavli Neuroscience Discovery Institute. We're excited to share our results in developing biological and computational tools to visualize millions of synapses in living mice to explore how the brain encodes behavior. A central goal in neuroscience is to understand how information is encoded and stored by the brain. A well-studied model of information processing is synaptic plasticity, in which regulation of AMP receptor trafficking dynamically modulates synaptic strength, either potentiating or weakening specific neuronal inputs. While we ultimately want to understand how synaptic plasticity drives behavior, studying these tiny and extraordinarily dense structures in living mice is extremely challenging, as you can appreciate from these videos of our raw data, where we fluorescently label millions of synapses and visualize how they change over time in living mice. Today, I will share with you our progress in developing and applying computational methods to automatically track synaptic plasticity underlying behavior. Our approach leverages tools from many fields, including molecular biology to genetically tag synaptic proteins, systems neuroscience to image these labeled synapses in living mice, and data science and computer vision to track how these complex synaptic networks change during behavior. To visualize synapse dynamics in living animals, we've made transgenic mouse lines with fluorescently labeled endogenous AMP receptors. This tag is pH dependent, enabling us to visualize only protein expression in the plasma membrane as fluorescence of non-functional internalized receptors is quenched. This transgenic line labels all GluA2 containing synapses, and we're labeling nearly every excitatory synapse throughout the entire brain on the order of millions of synapses in mice. Indeed, using in vivo 2-photon imaging of these SEP-GluA2 mice, we were able to discern millions of individual synapses within small subregions of neocortex, as we see here in one plane of SEP-GluA2 in neocortex. Importantly, we're directly imaging AMP receptors whose relative expression level determines postsynaptic strength. Indeed, I've previously validated that brighter SEP puncta correspond to functionally stronger synapses. So, using these tools, we can track plasticity with unprecedented scale and detail, exploring the synaptic architecture of behavior with single synapse resolution across entire brain regions in living animals. While this has the potential to significantly advance our understanding of the molecular basis of behavior, interpreting these unprecedentedly detailed datasets requires new computational tools to automatically track how synapses are changing over time, as manual annotation at this scale is simply not feasible. To facilitate the process of tracking millions of individual synapses over time during learning, we use a deep learning approach called CARE, or Content-Aware Image Restoration, to improve the effective resolution of in vivo 2-photon microscopy. Very briefly, CARE networks are trained using optimally imaged tissue taken using super-resolution area scan confocal microscopy as ground truth. We then generate training data using aligned and registered tissue that has been taken using suboptimal imaging, achieved by changing the laser power, exposure, scan speed, etc. We are then able to apply this neural network to effectively enhance the resolution of otherwise noisy in vivo 2-photon imaging in behaving mice. The novelty of this approach is that CARE is able to restore optimal area scan level resolution to otherwise noisy in vivo images in behaving mice. You can appreciate the power of our application of this neural network by comparing videos of the same in vivo volumes before and after applying CARE. Again, CARE beautifully restores optimal resolution to these otherwise noisy in vivo images. Here's the pipeline we use to automatically detect, register, and segment millions of individual synapses in vivo. Briefly, we image the same volumes of cortex each day, align the daily volumes using elastics, apply the CARE restoration model, and automatically detect synapses using a watershed algorithm in ilastic. In the left video, we're switching between the CARE output and ilastic detections, which are rendered in arbitrary colors. And you can see how well we're segmenting hundreds of individual synapses in the single imaging plane. And on the right, you can see the richness of our 3D detections in the in vivo volumes. We've rigorously validated this network to convince ourselves that CARE output reflects real synaptic signal. As you can see in the bottom row, we find that restored synapses from in vivo 2P volumes closely match ground truth, and further that the restoration process doesn't distort the underlying synaptic signal. We see this on the right in the Jacquard index of paired, restored, and ground truth detections, which support the rigor and precision of our network, that the synapse detections aren't changing the underlying shape, density, and distribution that we see in the ground truth. In the middle, you can see our relatively low overall error rates compared to ground truth. We have a particularly low false positive error rate, and most of the errors that we are seeing are simply misses of small or dim synapses. With these tools, we're interested in tracking synaptic plasticity during behavior using longitudinal in vivo two-photon imaging of retrosplenial cortex during exposure to novel environment. In this experiment, we install cranial windows over cortex of Septglory 2 mice, and we image the same volumes of labeled synapses over several days and observe how synaptic plasticity changes both at baseline and following novel spatial experience. On the left, you see a depiction of the gross alignment of large brain regions for each daily imaging session. And on the right, you see a zoomed in example of fine scale synapse alignment. Aligned voxels are displayed in white, and I hope you'll agree that we're doing a remarkably good job of registering and tracking these rich synaptic networks, especially considering that we're able to track these submicron structures in living mice. So, what's happening to synapse dynamics at baseline and during novel spatial experience in these mice? To answer this question, we compare how each registered synapse changes over time. In the plots on the left, each row is a single synapse from a representative cortical volume. Synapse strength is normalized to its day zero intensity, so we can easily track relative changes in synaptic plasticity over time. Synapse warm colors denote an increase in synaptic strength, while cooler colors represent a weakening. While we do see some slight changes in synapse dynamics at baseline, increased and decreased synaptic strength seems to be balanced in these home cage control mice, suggesting a homeostatic mechanism. However, following novel spatial experience, we observe significant potentiation of synaptic strength, which you can see in the middle, consistent with the induction of long-term potentiation, a well-studied form of synaptic plasticity. Overall, these proof of concept experiments show that care processing and in vivo imaging can be used to track how changes in synaptic plasticity encode behavioral learning with unprecedented detail and scale. And with that, I'll conclude by thanking our funding sources and you for your attention.